Hi everyone, welcome to our talk on ranking of communities and multiplex spatiotemporal models of brain dynamics. I'm James Wilsenach. I'm currently a postdoc at the Wellcome Center for Human Neuroimaging, and this is work that I did as a PhD student at the University of Oxford, working with Katie Warnaby at the Wellcome Center for Integrative Neuroimaging, and Charles Dean and Gesine Reinhardt at the Department of Statistics. So first things first, I just wanted to go over a sort of a general framework that is quite common in neuroscience and cognitive neuroscience, which is this notion of static state models of behavior. So people might be familiar with characterizing certain behaviors or certain modes of behavior in terms of networks. So these are things like the resting state networks that most of us will be familiar with, and that this is good and well, and it sort of has a place, and it produces, we expect that under a particular resting state network, we expect certain behaviors that are visible, let's say in fMRI or EEG, that should be, sort of relatively common across subjects, maybe somewhat differ between them. So there's sort of a subject-specific expression of that thing, and then there's a general sort of pattern that particular resting state network evokes. But we also know that there's a dynamic reconfiguration of the brain during both rest and task. So even at rest, the brain is constantly reconfiguring to perform certain tasks, whether they be housekeeping tasks or tasks that are sort of in response to a specific stimulus. And so we think that sort of dynamic state models capture more of that transitional reconfiguration that the sort of static standard network model doesn't really do. And we characterize these sort of reconfigurations, or one of those configurations, as a community, where communities sort of organize these activities in both sort of time. So you can here see a model, a dynamic model of different brain states and the transitions between them. And here we can see sort of an organization of those states into a temporal community of different states. And then also within those states, between the different brain regions, here we've got sort of a cartoon of a brain with maybe 6 different regions. Then we can see different configurations of those brain areas and their coordination between them as sort of spatial communities. So temporal communities and spatial communities. Right. So how do we model this sort of level of complexity, this sort of spatiotemporal level of brain dynamics? One method that's been proposed and quite widely used is the hidden Markov model. So hidden Markov models are quite useful because they can, in an unsupervised way, or relatively unsupervised way, find estimates for both the state activity, so the activity of the brain in a specific state, and it can sort of segment the time series of brain activity into various states. So here we have sort of the transition dynamics, which is the probability of state moving from state S to state S dash, given that you're already in state S, and the activity, so we could call the functional activity, so the expected correlation between different brain regions and the sort of expected mean between different brain regions, given that you're in state S. So we take this one step further, and we consider what we call the hidden Markov graph model, which takes as layers, so it's a graph, it's a multi-layer graph, and it takes as layers state activity, and as in between links, it takes the probability of transition between states. So here you can see an example of such a graph, right? This is a graph that sort of generalizes this notion of both within state dynamics, so what's happening at the level of mean activity and covariance of activity, and then the transitions between different states. And this gives sort of a more complete picture of what's happening from from time t to time t plus 1 within the brain. So we've discussed this notion of community dynamics, where different brain regions can belong to spatial communities, and we have transitions between different brain states that encode certain relationships between brain regions. And in the temporal case, it's relatively easy to find these communities. There are many different methods. Innovinc community detection is one, linkage community detection is another, but there are many different sort of network or graph-based methods for finding these. In the spatial case, it's a bit more tricky because now we're involving all of the different possible brain regions. And so there's sort of a combinatorial issue of having lots and lots of different possible brain regions that could combine to form a spatial community. And also we're dealing with a dynamic model, so we prefer that the dynamics that we come up with, or rather the communities that we come up with, are sort of temporarily plausible. We wouldn't want one of the communities to be a radical reconfiguration from one state to the next. And the other problem is that there's really no obvious metrics for weighting one particular community and saying this community is sort of more true and more representative of the particular state than any other. So we've got a proposed method for doing this, for performing community ranking, to find the most sort of plausible and most representative communities of each state. And the way we do this is using the HMGM structure itself. So first we use a scoring function that finds what we call the functional homogeneity of each community. So how much functional agreement there is within a community, a spatial community. And then we sample from the, remember the HMGM itself is a probabilistic model. So we sample from the HMGM plausible possible spatial communities, both within and between states. And we compare those sort of plausible communities to the one that we actually have using whichever community detection method we choose, and then validate these against Neurosynth, which is a meta-analysis tool that takes in a large corpus of cognitive and sort of behavioral neuroscience data in the form of literature, and then analyzes that literature to come up with terms that are associated with certain locations in M&I space. So it'll come up with a scoring for each point in M&I space, and then if you input a particular activity map, it'll output the most plausible associations between that activity map and possible functions such as vision, or possible tasks such as visual tasks like discrimination tasks, for instance. And so Neurosynth can be used as a sort of alternative or a possible other way of measuring the functional homogeneity of a particular spatial community, a particular set of brain regions. So looking at our results, we applied the HMGM to a small cohort sort of at baseline, so resting with eyes closed, and found that sort of in support of previous literature, really, the HMGM is both modular, so the structure of the model that we get out is sort of modular in the way that previous results have shown that, activity at rest is generally sort of performed in these functional modules, and hierarchical. So there are certain states that are more important than others. And here you can see the breaking up of these states into various modules across, I think it's 19 states. Some of them have been taken out for being too brief, which is part of the filtering procedure that the HMGM performs. And then from each of the hub states, we did an analysis to find out what the, using Neurosynth, we did the analysis to find out what was the sort of dominant annotation, or what was the sort of dominant function of each of those communities, the spatial communities now, that were highly ranked. And we found that, usually they made some sort of functional sense, that maybe the subject was hearing something in the room, or that there was some deeper functional process, maybe a homeostatic process that the brain was going through while at rest. So, sort of in conclusion, we have produced a method for data-driven that is sort of entirely unsupervised. We don't use any supervised learning in our pipeline, so you can input your fMRI or fMRI time series, and from that produce a long model that will potentially sort of summarize the entire dynamics of your subject in a particular condition, and then break those different activity patterns up into communities that we think summarize the dynamics in either a particular state or across states. So our study, though, was sort of a proof of concept using a fairly limited cohort. I think it is useful to note that we were able to train a model that sort of came up with coherent results, that was coherent even though it was a very small subject pool, very small cohort. And we proposed that also in looking at the literature and at looking at the sort of the way that dynamic models are discussed, there needs to be sort of a shift away from this sort of static view of networks and more of a dynamic sort of modular view that encodes the sort of important sort of new terminology that's needed to describe the brain not at rest or not in a specific configuration, but transitioning between different configurations. So we need to come up with sort of a more dynamic terminology set, and we propose that maybe the use of the word community can help to bridge some of that gap. Also, during our sort of analysis of the neurosynth tool itself, so this meta-analysis tool that takes in this literature of this large literature cohort, we found sort of structural biases that might be an issue when you're analyzing these sorts of data. So when you're analyzing, let's say, even just a static fMRI image or a static analysis that you've done, let's say using SPM or some other software tool to perform sort of static analysis of different activity maps, and we find that there's somewhat of a bias within the cohort itself towards sort of common tasks that people tend to perform. So things like vision sort of predominate as being highly active or highly cited and highly used as a term to describe tasks in the sort of cognitive and behavioral neuroscience literature. And we are now working to apply the HMGM framework to understanding unconscious dynamics. So not just sort of resting state dynamics, but now under specific conditions. And I'm happy to share some of the details of that work if people would like after the talk. Thank you.